All right, so this lesson is going to be on the process called meiosis, which shouldn't be confused with mitosis, which is what we covered in some of the previous videos that I've done. So meiosis is a very special process that instead of doing like mitosis, where you make a copy of the DNA and then it splits in half, and you end up with one original copy in each. This actually involves a replication and then two divisions. So if you start with a certain amount of DNA, you copy it, you double it, then you divide it, you go back to kind of a normal amount again, and you divide it again to get half of what you originally had. That's basically what meiosis does. Plus, what it also does is it creates genetic variation through a couple different mechanisms, which we'll talk about, with the inevitable goal of creating gametes, which are sex cells or sperm and egg. All right, so first, again, we'll review sexual reproduction. Remember, asexual reproduction only uses one parent, where sexual has two parents that provide two different sex cells, either a sperm or an egg. The female makes eggs, and males make the sperm, and they fuse together and eventually form a full organism. Now, why are sex cells important? Remember, in body cells, chromosomes are found in pairs with each other. Certain pairs pair alike depending on the genes that are on them. Okay, a cell with two of every kind of chromosome, so meaning they have two of the same type of chromosome, is called diploid. So they have two copies of every gene, though sometimes the versions of those genes will be a little bit different. If a cell only has one copy of every gene, or one type of every chromosome, that's called a haploid cell. And sperm and egg are considered haploid cells. So in a human, we have 46 total chromosomes in our body cells, or they're called somatic cells. In our sperm and egg, we have 23 in each. So when you fuse a sperm with an egg, we get 46. Now, the sperm has a full set of chromosomes in it, and the egg has a full set of chromosomes in it. Okay? But remember, in us, we're diploids, so we have two full sets of everything. One set from mom, one set from dad. All right, so sexual reproduction starts with two sex cells, and each are haploid, as I said, and the process ends when the two sex cells fuse, which is called fertilization. The zygote is formed, and that zygote, which is going to eventually become all of the cells in your body, is 2N, or diploid. So if we take an N sperm, a haploid sperm, plus an N egg, which is haploid, we put them together, we will get a 2N organism, or something that has two copies of every single gene. So this is kind of showing you homologous chromosomes and what I mean. Now, these two chromosomes, they have the same genes on them, but sometimes they have the same version of genes, such as in this case where this red matches up with the red. Okay, so it could be, for example, this could be the brown-eyed gene and this could be the brown-eyed gene. They both have the brown-eyed gene. Whereas up here, maybe we have two different versions of the same gene. This is the hair color gene, and this is the brown hair color gene, and this is the blonde hair color gene. So that's what we mean by different versions of the same gene. Now notice here, these are two homologous chromosomes and these are two homologous chromosomes. Together, these are two identical copies called sister chromatids. The sister chromatids form together during and after DNA replication. So the DNA would replicate, we have two identical copies of the genes. They'll then condense into chromosomes which actually stick together. So those are identical copies. Remember, homologous chromosomes and sister chromatids are not the same thing. Each one of these is a sister chromatid, okay, with these two being sisters and these two being sisters. But homologous chromosomes are different chromosomes, but they have the same genes on them, but sometimes different versions of the gene. And you got one of those chromosomes from mom, and one of them from dad. Think of them almost like how if you have a pair of anything, like a pair of shoes. Your left shoe and your right shoe, they're not exactly the same, but they come in a pair. That's how you want to think of homologous chromosomes. So, as we said before, meiosis generates sperm, an egg, called sex cells. The process of meiosis itself is not sexual reproduction. It's the process of making the cells that we will use in sexual reproduction. The goal of it is to reduce chromosome number by half. So if we start with 2 N cells in our body, we're trying to make N cells in meiosis, excuse me. Another important thing that meiosis does is it generates genetic diversity. And we'll talk about the mechanism behind how it does it. There's actually really two ways that it happens within there. But genetic diversity is super important for the overall survival of the species of that organism. Okay, so this is how you essentially want to think of meiosis. We start with, here are two homologous chromosomes right here. So they're the same size, have the same genes on them, but sometimes different versions of the gene. What we're going to then do is we're going to replicate those chromosomes just like we did in mitosis in S phase. Now we have two sister chromatids, here and here, with two homologous chromosomes. So each homologous chromosome has two sister chromatids. What we're then going to do is we're going to split apart those homologous chromosomes in our first part of meiosis. Notice how they've been split here and here. And then we're going to divide again without replicating, and we end up with one chromosome in each new sex cell that we've created. So notice we start with 1, 2 of the same type. That's where we get 2N from. And in the end, we end up with only one of each type. That's called N, or haploid. The process of dividing sister or homologous chromosomes is called meiosis 1. The process of dividing the sister chromatids in half is called meiosis 2. So here's showing you the process, in a little bit more complicated way than we just saw. Here's the chromatin. It condenses into chromosomes. The difference here, though, so this is showing meiosis 1, which is the first process where the goal is to separate homologous chromosomes. You can actually see the homologous chromosomes pair up with each other. That didn't happen in mitosis, but in prophase 1 of meiosis that happens. You want to remember that. Prophase 1 is where the homologous chromosomes will come together. They then do this process called crossing over. Sometimes they'll call it synapsis. But they form this structure called the chiasma, which is where the chromosomes kind of cross over each other, and the two homologous chromosomes will actually switch pieces of them, basically mixing each other up. So for example, one of these chromosomes came from your mom and one came from your dad inside of you. What this is doing is it's taking your mom chromosome and dad chromosome and jumbling them together. This is one of the first places that we create genetic diversity in meiosis. So in this process, we're crossing over different genes and chunks of the genes, which will actually put different pieces on. As you can see here, some of the blue has some red on it, and some of the red has some blue on it, to create genetic diversity. Then during metaphase, the homologous chromosomes line up in the middle. Then the homologous chromosomes are pulled apart, and as I said, notice they're all genetically kind of unique now because we've mixed up the DNA in the crossing over process, which happened during prophase 1. Then what will happen is it'll do telophase and cytokinesis, and then it condenses the chromosomes back, or decondenses it to chromatin, but then they get condensed back into chromosomes again. Now these just have sister chromatids on them. Then those chromosomes are going to line up back in the middle again, like this. The sister chromatids will get pulled apart, and now we have cells that each have only one copy of each gene as opposed to two in our original cell. Notice how many cells we made. We made 1, 2, 3, 4. We started with 1. Okay? So what happened during meiosis 1? The homologous chromosomes are separated, and that process called crossing over occurred during prophase 1. And then during meiosis 2, the sister chromatids are separated, just similar to how that happens in mitosis. All right, so we talked about one way we increase genetic variation already with crossing over. There's another way, which is through a process called independent assortment or random assortment. We're going to talk about this again when we get into Gregor Mendel, but he actually came up with this hypothesis without seeing any of the chromosomes, just based on how traits were passed on to different generations. But this is going to show you only 2 chromosomes, for example, and how independent assortment works. But remember, we have 23 different pairs in us for a total of 46. So here's showing you 2 pairs of homologous chromosomes, And the idea is they can line up like this, or they can line up like that. So you get different combinations, okay? So if the homologous chromosomes decide to line up like this, regardless of the fact that we have crossing over occur, this will be the resulting meiotic products down here. But if they line up like this instead, where they just kind of flip-flopped, we get different meiotic products. Now, as I said, this is only two pairs of homologous chromosomes. Remember, imagine 23 different pairs. If you can do the math behind that, there's two different possibilities to the 23rd. There's over 8 million different genetic combinations just because, or just as a result of independent assortment, if we have 23 pairs of homologous chromosomes. So those are all the different combinations you can get. All right, this is going to show you again crossing over, because we're talking about what generates genetic variation. We talked about this already. Remember, during prophase 1, the homologous chromosomes will actually switch. Sometimes some people call it switching socks. I don't know how Ms. Bebeck talked about it. I think she did it with sandals or something when she taught about meiosis. But pieces of one chromosome are going to go on to the other one, and they're going to create recombinant chromatids, because it's called genetic recombination. It's another word that they use for crossing over that you just want to have in your arsenal, just in case they use that word on the exam. But they recombine, and they form these new unique chromatids and chromosomes, okay? And remember, the X shape that is formed is called a chiasma. This always blows my mind to think about this, but between independent assortment and the process of recombination or crossing over, no two sex cells have ever been created statistically. Now, there could have been, but statistically speaking, no two sex cells could have been created that were alike, ever. And there are more possible genetic combinations in human sperm and egg, than there are atoms in the entire universe. In all of existence, there's more possibilities. So that's why it's not really possible to have an identical twin through the normal process of sexual reproduction. You can have identical twins, but that's caused by one fertilization event, and then after it does mitosis, the eggs split into two different individuals, so they actually are clones of each other. So the differences between making sperm and egg. They go through the process of meiosis 1 and 2, both of them. The only difference is in sperm, we actually create those 4 cells at the end, and all those cells become sperm. But in eggs, that's not the case. In eggs, after every meiotic event, one of the eggs actually shrivels up, and it becomes what's called a polar body. And then the other egg ends up doing meiosis 2, and then the result of that one becomes a polar body, and you only get one egg as a result of a meiotic event, in contrast with 4 sperm. And the egg is super important because the egg holds most of the nutrients, most of the mRNAs to get development going, and also holds all of the mitochondria. So all of your mitochondrial DNA that you have in your body actually came from your mother's lineage. So she got them from her mother, and she got them from her mother, and so on and so forth. There are other modes of reproduction that we've kind of talked about already. Some of them we did, some of them we didn't. Conjugation is actually bacterial sex. And in conjugation, what a bacteria will do is it'll actually send what's called the sex pilus out, and that's called the positive bacteria. And it'll send it out to a negative bacteria, which will receive the sex pilus, and it'll just take its DNA and it'll send it across and swap some of its DNA with the other one. So it's kind of like increasing genetic variation in bacteria. They'll pass plasmids and things like that, which we talked about which are the small circular pieces of DNA that bacteria sometimes have and can pick up. So that increases genetic variation in bacteria. It's not true sex. It's not like meiosis and doing the whole sexual reproduction process, but it's more similar to what they normally do. We talked about vegetative propagation in plants previously, where you just send shoots out where they're exact copies of each other. But what we haven't talked about is this really neat thing called parthenogenesis that happens in animals. Only certain animals do this. Whip-tailed lizards are an example of this. I think maybe the Komodo dragon can do it as well. And essentially what a mother will do is, instead of producing an egg that needs to be fertilized by sperm, she creates a haploid individual. So she actually births or lays eggs that are haploid, and those haploid organisms become, or those haploid eggs become organisms, and then those organisms can create haploid organisms and so on and so forth. That's called parthenogenesis. Basically, they're making clones of themselves through laying eggs and not doing fertilization. Last thing I want to kind of go over in this lecture is karyotypes. Now, karyotypes are basically if you take out chromosomes, and we're going to talk about karyotypes again actually, but if you take out chromosomes and look at them and pair them up based on with their homologous partners. So we can see we have 23 pairs total, 1 through 22, plus a set of sex chromosomes down here, X and Y. We have two X's here. If you're XX, that means you're a girl. If you're XY, that means you're a boy. So if you look at this karyotype, you could say, oh yeah, it's a girl, because it's XX. But notice, these are homologous chromosomes, similar banding pattern, similar length. And these are all arranged by size. Chromosome 1 is the largest. Chromosome pair 22 is the smallest. If you've ever heard of trisomy 21, which is Down syndrome, that means instead of having only 2 copies of this, you would have 3 here, or at least a partial part of a third. But we'll talk about that more when we get into chromosomal abnormalities later in the course. Here's showing you the same homologous chromosomes, but they have replicated. So these are now copied versions. In both cases, now this is a common misconception, this is diploid. It's diploid because it has 2 of each type of chromosome. This is also diploid. This is not considered 4N. This is diploid, but each homologous chromosome has a copy attached to it. So each homologous chromosome has 2 sister chromatids. Remember, those will get pulled, the homologous chromosomes get pulled apart during meiosis 1, and then during meiosis 2, the sister chromatids get pulled apart. And notice this is a boy because it has an X and a Y. Here's showing you the karyotype of a human sperm. Okay, 1 of each type. The dad in this case is giving a Y. And then in an egg, 1 of each type, the female is giving an X because that's the only thing she can give. And then when they fuse together, they form a full karyotype that is diploid. And in this case, we made a boy. We will talk about sexual reproduction later in the course, more in depth, how an egg develops. But remember, you take a sperm from a male, which is N, or haploid, and an egg from a female, which is N. You fuse them together, you create a zygote, which is diploid, or 2N. Each zygote will have 2 full sets of chromosomes, 1 from mom, 1 from dad, and that's why it's called diploid. And what we're going to do next is we're going to talk about the implications of this process in the production of traits in our bodies.